All right, let's get going. So this is the second lecture of Unit 3, T-cell development. So a very dense lecture, kind of like we had with the B-cell development lecture, but I don't think it's too much. Now, obviously, I would say that, but just bear with me. I think we'll be okay. A lot of good content that we can apply to later physiology and later diseases, so it's cool to learn. And thankfully, we have some familiar terms here. So the hematopoiesis, of course, is in our bone marrow, the blood cell generation, so a little bit from last unit. Our T cells are going to form, just like the rest of our blood cells, from that hematopoietic stem cell. And that can become a common lymphoid progenitor. If we're talking about the T cell pathway, then that's the pathway we're going to choose. So the CLP can become a B, T, NK cell, or a dendritic cell. Cool enough? But in this case, we are obviously talking about T cells. T cells. So I'm going to commit to the T cell lineage. And when I do that, I'm leaving the bone marrow. Get out of town. So HSC, CLP, early T. Once I decide I want to become a T cell, leave the bones, go to the thymus for college. So chemotaxis and entry, those progenitors leave the bone marrow, enter the blood, and home to the thymus. I really can't explain how they do that. I think it certainly is a chemotaxis, a following of chemical signals and gradients. But I'm sure if I were to explain it to you, it would sound like magic, because it sounds like magic to me. So all you need to know is that once they leave the bone marrow, they home to the thymus where they are educated properly. Again, from last video, we have the cortex, which is the outer region of the thymus, this darker orange that I have here. And that's going to be densely packed with cells and developing thymocytes. That's where that blood-thymus barrier is at its most robust. Then we have the inner nougat, the medulla. Fewer cells, much more dispersed in this region, and it's a site of negative selection. So remember, that is testing for autoreactivity. So in this case, in this region, the blood-thymus barrier is not as robust. We actually want a little bit of self-exposure, because if those T cells that we've made are autoreactive, well, we need to know that and do away with them. So thymic education, making naive, mature T cells that are functional and self-tolerant, that tolerate the self. We don't want to be mean to the self. So the goal of T cell development is making a functional CD4 positive T helper cell or a CD8 positive cytotoxic T cell. I said I was going to do my best to eliminate receptors and algebra-looking stuff from the class, but there's some that I cannot remove, and there's going to be a couple this unit. I apologize in advance. These are certainly receptors that I cannot ignore. The CD4 receptor is what makes a T helper cell, and same for the CD8 receptor, making a cytotoxic T cell, So make sure you associate these receptors with their pair. We call T helper cells CD4 T cells. And same, CD8 T cells are your cytotoxic T cells. So that's the goal. We want to make these guys. But how do we do that? Let's see the stepwise process here. Again, you have the freebie. If you remember and studied your hematopoiesis well, you know we start with a hematopoietic stem cell. Then we go down the common lymphoid progenitor pathway for my B, T, NK, and dendritic cells. And then here's where the new terminology comes along. We become a double negative T cell while we're in the thymic cortex. No CD8 and no CD4. Okay, think about like you just graduated high school and you're just like, man, I got no idea what I want to become. No earthly idea. So I'm not expressing any receptors. I don't know what type of T cell that I want to set out to be. And then during this process, we have what we call beta chain rearrangement. If you will hearken back to last unit where we had the heavy and light chain of the antibody, the B cell receptor, well, we are going to have a similar process here. we have to form, not only do we have to decide what T cell we want to be, but we also need to make our T cell receptor. And here he is right here. Less unique and cool looking as the double-barreled Y that the B cell receptor has. This is just 2 lines, so it's kind of boring, but this is your T cell receptor, made up of a beta chain and an alpha chain. The top of it is the variable region, so that confers that antigen specificity. And the bottom is the constant region, so that's just going to stay the same and anchor the receptor to the cell. So a little bit of extra layers here. I have to make sure my T cell receptor is functional, and also I need to add either a CD8 or a CD4 receptor so I can decide what T cell that I want to be. So I'm giving you, hopefully you handled the steps of B cell development well, because we just focused on the B cell receptor there. Now I'm giving you 2 receptors to focus on. So just building and building on the foundations that we've set thus far. So I'm naked when it comes to CD8, CD4, but I'm working on my B cell receptor. Or I'm sorry, my goodness gracious, my beta chain of my T cell receptor. Gosh. All right, the next stage is a double positive T cell. So I'm expressing both CD8 and CD4. So I like a good parallel for this is if you're thinking med school, a lot of folks say, you can have the specialty that you like, in your head coming in, but know that you're going to be wearing multiple hats, right? That's the 3rd year. You do a bunch of rounds in different clinics. You're going to hit general surgery. You're going to hit OB. You're going to hit everything under the sun. So there are aspects of your collegiate life and of your beyond collegiate life where you wear multiple hats. You don't know what you want to be just yet. You're figuring it out. That's the double positive T cell stage. I've got both hats. I'm expressing both receptors because I'm not sure what I want to be just yet. During that process, also, is when I finish my T cell receptor. I bring in the alpha chain, rearrange that around a little bit, and finish forming this big old guy right here. After that, then I move on to the single positive T cell. The single positive T cell, so that's exactly what it sounds like. Single positive. So I'm only expressing one of my identity receptors, either CD8 or CD4. Remember, CD8, single positive, means I'm a cytotoxic T cell, while CD4 positive means I am a T helper cell. So make sure you link those together. And now finally I am a mature, naive T cell. Remember, mature, fully developed, but still naive. Still naive. The only thing that makes it not naive is when it's activated. When it's activated is when the T cell's specific antigen has been presented to the T cell. So still naive. You'll also notice that I have the checkpoints laced here, exactly like the B cell receptor from last unit. After I finish forming my T cell receptor, my beta chain is done, my alpha chain is done, I check and say, hey man, you good? Can you work? Can you do what I need you to do? Are you okay? That's the first checkpoint. I just finished piecing it together. Does it work? Then after the single positive stage, I test it out. Do you bind to me? Are you autoreactive? If so, get the heck out of Dodge. I don't want anything to do with you. Autoreactive T cells, not good, not helpful for my health. All right, so those are those 2 checkpoints, both occurring in the thymus. But let's delve into those stages a little bit more, give you a little more clarity. So again, that double negative T cell means no CD4, no CDR. I don't know what I want to be. Don't know what extra surface proteins I want to express. And during that, we are rearranging those T cell receptor genes. In this stage, it's the beta chain only, so the yellow. If we get a successful beta chain, then that makes a pre-TCR. So I got half of it working. During this, before the alpha chain gets ready to go, there's like a placeholder here. Don't worry about that, though. Just worry This is the beta chain stage rather. If we get an unsuccessful beta chain, then we just kill that cell. We have no need for a poorly functioning T cell. So that's what we look like. Our double negative is here. Our beta chain is on the up. That's what the arrow is. And I'm saying the alpha chain is just kind of being held up together by a placeholder right now. It's not fully functional. And that gives us a pre-TCR, that B cell, or I'm sorry, that beta chain working and developing while our alpha lags behind. Then we have our double positive T cells. So this is the med student with multiple routes that they can take. The one with all the potential. We can take the CD4 path or the CD8 pathway. Double positive refers to having both of those being expressed. So it's the most abundant population in the thymus. So I think that harkens back to my example of the thymus being a site of education, right? That's where you're finding yourself, finding what kind of employment or what kind of things you want to learn. And while you're doing that, you learn a bunch of different things. You know, like when I was starting out, when I was in the undergrad level, the amount of different types of biology I learned was vast because I didn't know what I wanted to specify into yet. So that's what's going on here. We have CD4 and CD8 expressing the multiple pathways we can go down. Also here, alpha chain rearrangement. We bring in the red side. So now this pre-TCR is done. Now it's a TCR. We have both the alpha chain and beta chain here. But now it's formed, I got to make sure that it works. So in the thymic cortex, so the outer region, the orange region that I had, does the TCR recognize MHC? This is how we test if it works. We're going to talk a lot about MHC later down the road, but remember, this is the self marker. So this determines if we can this determines if it just binds to the receptor that it needs to. Does it work okay? Are you good? If it can't bind MHC, if it can't recognize this protein, then we get rid of it. If it binds it weak or moderately, then that's totally fine. But if it binds it too hard, then that's kind of a signal of autoreactivity. So with this step, we want more of a Goldilocks zone. We want not too little, not too much, just right. Our MHC is kind of going to act like a presenter, like a platter, if you will. And it's going to hold up antigens that we've come across. We want those T cells to bind the antigens that MHC presents, but we do not want it to hook onto MHC and say, I want this instead. That's bad. That's a sign of autoreactivity. So there's a balance to be had here that we'll talk about as we go through MHC on 3.4, so in 2 videos. But here we have that double positive cell. TCR's done. It's passed the first checkpoint, testing binding to MHC. And during this process, I still have CD4 and CD8. So that's double positive. I have both of those receptors expressed on my membrane along with my T cell receptor. Then depending on how the MHC binding went, if it liked one type of MHC or the other, it will select CD4 or CD8. So just depending on how that reaction went down. Again, we'll talk about that soon. If it liked CD4 more, then it's going to keep CD4, and now I'm a single positive T cell. So good, I'm getting close to finishing the process. All right. Let's bring it to this diagram here. This goes through everything in a nice fashion, hopefully not overwhelming at first, but let's go through it. So there's the thymus, and I'm going to zoom in here. So the orange region is represented up here. This is the outer cortex, and the purple down here is the medulla, the internugate. early T cell, that's like at the just left the bone marrow. Then we get to the thymus. Double negative. I don't know what I am yet. I'm not expressing anything. My T cell receptor isn't even ready yet. But at the end of this stage, my beta receptor is starting to pop up. So I have a little beta hanging out. Then at the double positive stage, I'm expressing both CD8 and CD4. And at this stage, my alpha receptor comes in. So my T cell receptor is done. Cool. So then after here, now I go through positive selection. I got to test if that B cell receptor, that T cell receptor works okay. So now I bring in my supportive cells of the thymus. Here is a cortical thymic epithelial cell. Remember, because I'm in the cortex right now. And those cells are going to express, we have MHC1 over here and MHC2, the different types of self molecules. The double positive T cell is going to check out MHC1 and MHC2 and determine which one it likes the most. And that will determine if I'm CD8 or CD4. We'll talk about it more on the MHC day, but the CD4 cells are going to like MHC2 more, and the CD8 T cells are going to like MHC1 more. So depending on how that binding went, we'll determine which one we like. MHC1, MHC2. But again, we have a whole day on that, so don't stress. Now, after we decided we were single positive, we go on into the medulla, and we have the medullary thymic epithelial cells. Now, these also have MHC1 or MHC2 and MHC1 hanging off of their membranes, but they have something else going on here. So let's look. So now we are in negative selection territory. So the medulla is going to handle determining if we're self-reactive. The medullary thymic cells can express something really cool here. Kind of marked their nuclei with this blue. And that blue, they can encode an enzyme called AIR. AIR is autoimmune regulatory enzyme. Go ahead and change that to blue. So it's a gene that gets expressed that codes for this enzyme. And this enzyme is so cool because it allows those cells, oh my gosh, I have that's the old abbreviation for those cells. Let's go. So the cells of my medulla are the MTECs. My MTECs. So the MTECs now use that AIR gene and they can express proteins from all around the body, which is so cool. They can express like insulin, myelin, that's what covers your nerves, and proteins from your thyroid, right? So basically you just get like a microcosm, a little bubble of your various bodily tissues. And not to mention, if you remember, the blood thymus barrier here is also a little weak. So we might be exposed to different things in the blood as well. All of this is the goal with this is to ensure that these T cells are not self-reactive. We want to make sure that the antigens that we express here, if it's from our own tissue, that it causes no reaction. We don't want to have T cells that bind myelin, right? That's what causes MS. We don't want to have T cells that bind antigen, or that bind, criminy sakes. We don't want to have T cells that bind insulin. That's going to create diabetic conditions. So it's really important that the AIR enzyme presents a wide variety of self-peptides that we can familiarize these learning T cells to. Once they pass that stage, boom, now I've reached maturity. So all good to go after I've cleared that autoreactive stage. Final steps, we emerge as a single positive, mature, naive T cell. So again, that can either be a CD8 cytotoxic T cell, or a CD4 T helper cell. And the destination is the same as the B cells was. To the spleen, the lymph nodes, the malts, just a diffuse antibody coverage or diffuse adaptive coverage throughout the body. What's really crazy about this process, though, and where the college metaphor kind of dies, of all of the T cells that enter, over 95% do not survive the selection process. That is crazy to me. Crazy. So we're very selective, and this process is very difficult to pass. So if you think about that, go back to this diagram here. That means the super duper majority of these cells, either their T cell receptor doesn't work and doesn't recognize your own MHC, or they bind your MHC and your self-antigens way too strong. So that is why it's such a selective process here. We want to make sure that these T cells are not autoreactive and that they bind correctly, because this is how we get really excellent adaptive defense. We vet these cells very, very roughly here. All right. Let me think here. Yeah, that's it for T cell education. Just to reiterate, though, want to recap things. It arrives in the thymus as a double negative T cell. We start the T cell receptor with the beta chain. Then we express CD8 and CD4 along with our developing T cell receptor. Once that T cell receptor is done, we go to the cortical thymic epithelial cell, where it binds and says, hey, do you like MHC1 or MHC2? And can you see the antigen that I'm presenting here? Can you see the antigen rather than the MHC complex? Do you react to the antigen more than you react to me? If it reacts correctly, then we pick which receptor we liked more, either CD4 or CD8, and now I'm at the single positive stage with a functional T cell receptor. But functional doesn't always mean perfect, so then we go down to the medulla. And there we have the medullary thymic epithelial cells, and these guys have the cool enzyme AIR, autoimmune regulatory enzyme. That allows them to produce self-proteins that are all around the body, like insulin, myelin, and everything. So they present a couple of those. They present some of those and say, hey, just so you know, you might come across insulin, you might come across myelin, and you need to make sure that you do not react to them. If they don't, good to go. We have a mature T cell. If they do react to something that the body makes, then we say, get the heck out of here. All right, so that's the process here. We have our two checkpoints that are carried out by our thymic epithelial cells. So that's the breakdown there. I hope that wasn't too dense or confusing. If it was, if you have any bumps in the connections there, please do not hesitate to let me know. Thank you, folks, very much for your attention, and have a wonderful day.